welcome to my channel, The Biology Station. As you know that in my previous video, I have discussed about two modes of asexual reproduction, that is fission and budding. In today's video, I am going to discuss about spore formation and its various types in detail. So let's get started. Spore formation. Spore formation is a very common mode of asexual reproduction in various organisms that belongs to the category of monorans, protista, algae and fungi. So spore formation is commonly seen in monorans, protista, fungi and algae. Right. As the name itself suggests, there will be a formation of spore. So before getting into the detail, let's define spore. So what is spore? Spore is a very tiny rounded body. Okay. It is tiny, somewhat rounded, single-celled, with thick or thin wall, light in weight, and dispersive asexual body or propagule. Clear? Other than this, once the spore falls onto suitable substratum, it germinates and develops into new individual. Develops into new individual. Right? So what are spores? Spores are very small sized, tiny structures. They are somewhat rounded or spherical. Okay. Single celled. They are covered by thick wall, majorly thick wall, but in some spores we can see thin wall as well. They are very light in weight, and because of that they are dispersive. Dispersive means they can be easily carried by the wind current or the water current to the distant places because they are very light in weight. So it's very easy for them to disperse to the different distant places and germinate as and when required. Right? And once this spore falls on the suitable substratum. Now what is substratum? Substratum is any solid suitable surface which is good enough for the germination of that particular spore. So whenever the spore fall onto the suitable substratum, it immediately connects itself to that solid surface and germinate and gives rise to a new individual. Right? Now, on the basis of motility, there are two categories of spores. On the basis of motility. Motility means ability to move. Whether the spores are motile or non-motile. On this basis, on the basis of motility, there are two categories of spores. So we have motile spores and non-motile spores. So under motile category, we have only one type, that is zoospores. Okay, zoospores are motile spores. Under non-motile category, we have four different types. We have Conidia, we have Chlamydospores, then there is Oidia, and the last one, Sporangiospores. Right? So let's discuss about the zoospores first. Zoospores, as I've already told you, are the special kind of motile spores. Now, why are they motile? What is actually making them motile? For that, you need to know about its structure. So let me draw the structure of zoospore. So zoospores are actually produced inside a sac-like structure. These spores are produced inside a sac-like structure, a complete covering, right? So inside this sac-like structure, there are present large number of zoospores, these structures. So they have a kind of a pear-shaped body or somewhat spherical sometimes. Okay, and each of them comprises of two flagella. Each of the zoospore has flagella. Then there is an additional structure here. Now, this sac is known as zoosporangium. Okay, zoosporangium is like a kind of a parent covering inside which the spores are produced. Okay, which spores? Zoospores. And this single structure is known as zoospore. It is known as zoospore, right? Let's see how single zoospore looks like. Let's draw the diagram of zoospore of Chlamydomonas. Chlamydomonas, as you have already studied this in class 11th, it's a kind of algae. Okay, now the zoospore of Chlamydomonas looks like this. It has a sort of pear-shaped structure. Then inside there is chloroplast, this portion. Inside there is a cup-shaped structure, right? This is a chloroplast. This structure is a nucleus. Here are present the contractile vacuoles, and somewhere from here long flagella, they come out. Okay, right? So let's do the labeling part. So this one is flagellum. It's a flagellum. Okay. These are vacuoles, contractile vacuoles. This structure, the cup-like structure, this is chloroplast. And this red colored dot which I have drawn is actually present inside the chloroplast, and this structure is known as pyrenoids. Pyrenoids, they are meant for the storage of starch. Okay, after photosynthesis, they store starch. Then this particular part, this is all filled with cytoplasm. It has cytoplasm, and this structure is a nucleus. Right. So this is about the structure of zoospore of Chlamydomonas. Clear? Let's discuss the features of zoospores. First of all, as I've already told you, that as you can see that zoospores are produced inside a covering or a sac-like structure. This parental sac is known as zoosporangium, right? So the first point will be zoospores are produced inside a sac called zoosporangium. Because of this reason, it is considered that zoospores, they have endogenous origin. Endo means they are produced inside something else, right? They are covered by something. So they have endogenous origin. Endo means within something or inside something, right? They are produced inside zoosporangium. That's why they are having endogenous origin, right? Then, secondly, as you can see in this diagram, it's clearly visible that there are two long flagella, right? Because as you know, wherever, in whichever structure we have flagella, that structure is considered as motile. It's movable, right? So because of these flagella, these zoospores are capable of movement. They're able to locomote, right? They can move from one place to another. Clear? So zoospores are motile spores because they have, or as they have, flagella. Okay, as they have flagella, or they have two flagella, right? So zoospores are able to move. They are motile because they have two long flagella, right? As zoospores are able to move because of flagella, so it's very easy for the zoospores to swim in the water if they are in an aquatic habitat, and to disperse to the distant places easily, right? So thirdly, flagella help zoospores to swim in aquatic habitat and disperse easily, right? So they can disperse to the distant places, and whenever they will find a suitable substratum, they'll germinate there and produce a new organism, right? So other than the flagella, there are a vacuole, there is a chloroplast, then there's a nucleus at the center. Inside the chloroplast, there's a starch storing body we have, that is pyrenoid. Okay, so this is the structure of zoospore of Chlamydomonas. Clear? Now, other than this, zoospores are most commonly formed in phycomycetes. For example, albugo. This is nothing but a type of fungus. It's a fungi, right? And zoospore is a very common mode of asexual reproduction in many algae. Right? One example I've already mentioned here, that this is your Chlamydomonas. Other than that, we have Alothrix as well. Alothrix is also a type of algae. So this is about the zoospore, which is a motile spore, only motile spore. Moving on to the non-motile spore. So let's talk about the first non-motile spore, that is Conidia. Non-motile spore. The first one is Conidia. As the name itself says, it's a non-motile spore, so there won't be any flagella present in them. And Conidia are the most common spores of Penicillium, a fungus. They are commonly produced by Penicillium. So they are produced by Penicillium, which is a type of fungus. Right. So let's draw the structure of Conidia. These are all of them. All these structures are actually different types of hyphae. As you have studied in class 11, that fungal body is made up of large number of filamentous structures, and each of the filament is known as hyphae. The group of group of hyphae, which actually forms the body of a fungus, is known as mycelium, right? So this is a structure of penicillium. These different types of hyphae are known as conidiophore, right? And at the tip of conidiophore, there are present rounded bodies. At the tip, they are growing rounded structures. Right. These rounded structures are nothing but the spores called Conidia. Clear? This is penicillium. Now, what are the features of Conidia? As it is clearly visible from the diagram, these Conidia are produced at the tip of a special hyphal branch called conidiophore, right? As they are present at the tip of the hyphae and they are not enclosed by anything. So that's why Conidia are considered to have exogenous origin. They are not endogenous because they are not growing inside, they are not covered inside anything. Rather, they are growing superficially, they are exposed to the external environment. They are not enclosed by any sac or any covering. So that's why they have exogenous origin. So the first point can be that Conidia are exogenous in their origin. Why is it so? Because they are present at the tip of special hyphal branch called Conidiophore. Right? Why they are exogenous? Exo means external, outside. They are present outside, exposed to the outer environment. So they are exogenous in the origin because they are present at the tip of the conidiophore. Right? As you can see that some of the hyphae they have single conidia and somewhere there is a chain of conidia. So they can be present or produced singly. Conidia can be present or they can be produced singly or as a chain of conidia. Right? Then as they don't have flagella, no flagella, and hence they are non-motile. They don't have flagella, so they are not able to move. Right? They just, when they get mature, they just pinch off from the hyphae and they are released as they are light in weight. They disperse with the help of wind current and they will find the suitable substratum, germinate there and produce a new hyphae or mycelium to give rise to penicillium. Right? So this is the structure of Conidia and some of the features of Conidia. Right? So moving on to the second spore, non-motile spore, that is Chlamydospores. Chlamydospores are thick-walled spores. They are thick-walled spores. Okay. And these spores are produced directly from the cells of a hyphae. Right? These are produced directly from cell of hyphae. Right? Hyphae, as I have already told you, a filamented structure which forms the body of a fungus. These are swollen. These spores are swollen because they store reserve food material and because they have very thick wall, they can withstand unfavorable conditions for a longer time. Right? Chlamydospores are mostly formed by agaricus, that is mushroom, and rhizopus, which is a bread mold. Both of these are fungus. So let's see how chlamydospores they look like. See, as I've already told you that these are produced directly from cell of hyphae. Now what are hyphae? Hyphae are the filamented structures which has large number of compartments and cytoplasm in each compartment or in each cell, and there is single or many nuclei as well, and these compartments are separated by this structure, septa. Septa separates the cells of the hyphae. This is hyphae. Right. Now, suppose if this cell of the hyphae convert itself into chlamydospores, then how it will look like? It becomes swollen and it will be having a thick wall. Right. So something like this is going to happen. A single cell of hyphae will be transforming into chlamydospores. So it will look like this. This is nothing but the hyphae. These are hyphae only. These are all hyphae. Right. But some of the cells of the hyphae, what they're doing is they get modified into a swollen structure after absorbing or storing large amount of reserve food material and acquiring a thick wall. Each cell of the hyphae transforms into a swollen structure, and this swollen structure is nothing but chlamydospore. Chlamydospore. Here also we have chlamydospore. This hyphal cell also got transformed into chlamydospore, and they have thick wall. They are swollen because they have large amount of reserve food material. They have large amount of reserve food material. As they have a very thick wall, so they can withstand the unfavorable conditions for a very long time, right? In mushroom and in the mold, these type of spores or chlamydospores are very common, right? So this is about the chlamydospores. Now the third type of non-motile spores are Oidia. These are not very common spores. So Oidias are formed from the hyphae only. Sometimes what happens is that the hyphae, the tip of the hyphae start breaking into tiny fragments, or the tip of the hyphae start getting pinched off from the rest of the hyphae in the form of small tiny fragments, and each of the fragment then transform itself into Oidia, right? These are thin walled spores. These are formed from hyphae, right? When they break into tiny fragments or pieces. Okay, so they are formed from hyphae. Under what situation? When these hyphae break into tiny fragments or pieces. Each tiny fragment, each hyphae or hyphal fragment develops into Oidia. They get transforms into Oidia. Oidia are most commonly seen in the case of agaricus, that is mushroom. Now let's draw the structure of Oidia. What happens is that, suppose this is a hyphae, these are hyphae. Now what happens at the tip of the hyphae, that these from this particular part, this part of the hyphae gets pinched off and separate from the parent organism. So they are released like this, they get pinched off and they form or convert themselves into Oidia. Right. So the hyphae itself break down into many or the numerous or multiple fragments, and each fragment further develops into Oidia, right? This is a kind of hyphae. This is about Oidia. Now the last non-motile spore is Sporangiospores. Sporangiospore are the thick-walled spores which are formed inside a sac-like structure again, right? And that sac-like structure is present at the tip of another set of hyphae. So let me draw the structure first. So this is stolon, type of hyphae, horizontal hyphae. These are root-like structures. These are nothing but these are another set of hyphae having root-like structure. These are known as rhizoids. This one is stolon. Then from the stolon arise a long, erect, stalk-like structure. This is another hyphae, the vertical hyphae, which is known as Sporangiospore. Now, Sporangiospore gives rise to a somewhat rounded structure. This structure is known as columella, right? And this columella is further covered by another round structure. This structure is known as sporangium. It is known as sporangium. Sporangium is the structure which is going to produce spores inside it, right? So large number of spores are formed inside the sporangium. So these spores are known as Sporangiospores. Okay. So this is the structure of a bread mold, that is Rhizopus, a fungus, that is bread mold. This is how a bread mold looks like, and the formation of Sporangiospore is the most common asexual mode of reproduction in Rhizopus. Right. Now, if this is a Rhizopus, it is, just suppose it is growing on a slice of bread. The suitable substratum for this type of organism is a bread, moist slice of bread. Right. Suppose it is growing on the spore will fall onto the slice of bread. It will germinate there with the help of rhizoid. It will first produce rhizoid. With the help of rhizoid, it will connect itself to the surface of the substratum. And then comes out stolon, and that gives rise to the vertical hyphae, that is sporangiophore. And from the sporangiophore, then at the tip arise sporangium. And inside the sporangium, we have large number of thick-walled sporangiospores. Right. So when the sporangiospores got mature, this columella, it start moving upward. It start moving a little upward. And when it start moving upward, it actually moves out of the sporangium. As a result, the sporangium splits open. It results in the dehiscence of the sporangium. Right. So from this particular portion, the sporangium breaks. When the sporangium breaks down, it opens up, it split open. The spores, they are released out. The sporangiospores, they are released from this particular space. Right. And these spores, on finding the suitable substratum, germinate and produce a new mycelium, which gives rise to rhizopus. Clear? So these are sporangiospore. They are thick-walled spores, and they are endogenous in origin. What is endogenous? I've already told you. Why they are endogenous in origin? Because they are formed inside a sac-like structure. They are produced inside a sac-like structure, that is sporangium. That's why they are endogenous in origin. Clear? So this is about the Sporangiospore. So this is about the spore formation, that is third mode of asexual reproduction. In my next video, I'll be talking about fragmentation and regeneration in detail. Now a quick reminder, do not forget to press the bell icon for my latest updates, and please like, share, comment and do subscribe to my channel, The Biology Station. Okay. Thanks for watching my video and have a great day.